Recording is on.
So again, good morning everyone who already joined. For today's session, this is the second session of the Module 1, Quantitative Fundamentals of this webinar series organized by the Young International Committee on Predictive Modeling in Food. And we are pleased to have Professor Tini van Boekel with us for this module, which is going to he already speaks about basic statistics yesterday, and today is going to lead us through a bit more advanced topic like regression. So before starting, I'm going to lay down a few housekeeping rules for those who were not here yesterday. So as you see, your microphone and camera are muted because we tend to be quite a lot. And to ensure clear audio for everyone, please keep yourself muted. If you have any question, just type it in the chat box. We'll try to collect them and share with the speaker. And if you have any technical issue, please let us know and we will try to assist you. Also, feel free to connect with us through the Slack community and our LinkedIn page. So I don't want to take any more time from Professor Van Boekel, so that he may have a few more extra minutes if you want to go through a few more concepts at the end of his lecture. So please, the floor is yours. I'll stop sharing so that you can share your presentation.
Yep. Is it?
Yeah, perfectly visible. Yes.
Yeah? Okay, good. All right, then I'll move this button a bit down. Right, so, well, also for me, a good morning if you are somewhere in Europe, or otherwise maybe in afternoon. As Federico already mentioned, this is part 2, in which we are going to build upon what we have discussed yesterday. And I will focus on a very important technique that is used a lot in predictive modeling, namely regression. So let's start. Just to refresh your mind, yesterday we discussed that it is very useful to characterize variation, which is all over the place. And the way to characterize variation is by probability distributions. And they are useful in two ways. You can show visually how observed data are distributed, so what the variation is in a visible way. But it is also a useful tool to visualize uncertainty, or if you want, the degree of belief that you have in certain phenomena, especially phenomena that are not observed yet. I gave the example of predicting the weather of tomorrow. You can do that, but you have no data, so it is a degree of belief. I also showed last time in part 1 that conditional probability is a very important concept, and we are going to use that today also extensively. And just to remind you, what is conditional probability? It is the probability of one event knowing that another event has happened or will happen. And the notation for this is listed here. It's the probability of A given that B has occurred or will occur. And we also discussed the two approaches that there are in the frequentist one and the Bayesian one, and that they are usually giving the more or less the same numerical results, but they are philosophically quite different. So that's what I also wrote down in this slide. The two approaches come to the same conclusion in terms of numerical estimates. But the frequentist approach gives only a point estimate, so one value, whereas the Bayesian approach gives a complete probability distribution. And yeah, perhaps maybe surprisingly, you could also say that the frequentist way is a sort of special case of the Bayesian approach, because if you use a very flat prior, which means that you have no information about the hypothesis, then you are exactly doing the frequentist approach, because that relies only on the data and not on the prior. But nevertheless, there is always this conceptual difference that point estimates in the frequentist way are always fixed. They cannot have a distribution by definition, because the uncertainty there is in estimating the data and not the values of the parameters. And confidence intervals, which you will always hopefully see in reports, are actually fictitious repetitions. It's what we imagine that would happen if we would repeat experiments over and over again. And this is not true for the Bayesian way, because there the distributions really express our belief, or if you want, our degree of uncertainty about parameters. And it's also good to realize that this posterior contains all the information that we have. Depends on the model and it depends on the data. And once you have such a posterior, you can derive many things from it. Now, why do I stress the importance of the Bayesian approach so much? Well, I see a lot of applications in food science, because in foods we have a lot of variability and a lot of uncertainty, and that makes it very well suited for a Bayesian analysis. And we talk about estimating parameters and regression in this part of the module. I will not talk about, in view of time, about model comparison and method comparison, because that is also very well possible with the Bayesian approach. We'll talk about uncertainty estimation. But when we look at the type of studies that are done in food science, then there are sensory studies, there are shelf life studies, there is risk analysis, there is human behavior studies like consumer behavior, and all of these aspects are very variable, in the sense that, well, people do not taste the same, people do not behave the same. So there will always be big variability. And once again, the Bayesian analysis is really very well suited to deal with. So another thing I would like to point out is that the Bayesian approach is really more or less expressing the way we think as humans. It is a sort of natural way to express our way of thinking. So therefore, it is also intuitive. And that is not true for the frequentist way. You really always have to think about what does it actually mean. And sometimes it's even counterintuitive, like a p-value, where most people think that this is a probability about the alternative hypothesis, which it is not. You always have to think, well, what does it actually mean? Another big advantage of the Bayesian approach is that you need much less samples than with the frequentist approach, because it not only depends on the data, but also on the prior. And basically what you're doing is updating. And so the posterior that you may derive today may be the prior for tomorrow. So this is also updating knowledge. This is also the natural way in which we think in science. And the focus is really on the probability of a hypothesis. It is not on what is significant or not. This should be a judgment of scientists and not of a statistical test. And this posterior distribution you can use, as I mentioned already, with a lot of further calculations in which you use the whole distribution rather than just one point estimate. So really, I think that the fact that we can now use the Bayesian approach with our computer power is really a development that helps us a lot. So this cartoon that I took somewhere from internet is showing that in a funny way. We go from, first of all, for instance, in the Middle Ages, science was not about data. Science was about thinking. So that was the Homo a priorius. Then in the 19th century, scientists also became to think about data. Um, and that became popular in the 19th century with what was called the modern universities, in contrast to the medieval universities where data were not fashionable. It was about thinking. Then in the 20th century, which we still learn today, is the Homo frequentistus, which means that we are all taught with the frequentist approach. But now we became more wise and we try to combine the data and the hypothesis, and we end up with the Homo Bayesianis. I thought it was a funny picture, so I just wanted to share that. I also want to mention that even though I'm much in favor of the Bayesian approach, it does not mean that the frequentist approach is wrong. It's perfectly correct, but the problem with the frequentist approach is that people do not interpret it well because they are not aware or just neglect the assumptions that are made, and that is a bit of a nuisance. So personally, I learned a lot from these books from a guy called Richard McElrath. He is an American, but he works in Germany. He's an anthropologist, and he has written well, two books basically with the same title, but one is the first version and the other one the second version, and they are both quite different. I learned a lot from this book, and I can really recommend it to you to update your knowledge. It's very nice, but also he has free online lectures which are really brilliant and very humoristic as well. So I gave you the link here where you can look it up for yourself. I can really recommend that you do that. It will take you some time because each lecture is 1.5 hours or so, and there are about maybe 15 or 16 lectures, I think. So it's quite a job, but they are very, very informative. I was very inspired by this book, so I'm also busy on rewriting my own book on kinetics, but that is a hell of a job, so I'm not finished with that. But I did publish some papers that make use of all that knowledge, and I will give you some references tomorrow. And if you want to have some more information, please feel free to contact me. Okay, that was just a sort of repetition of yesterday. I now want to focus on a very important technique, that is modeling and regression. So if you do modeling, at least in food science, you always need to confront those models with data because foods are so complex that we cannot just rely on theory. We need to measure. And then the question becomes, how can we extract the information in the data? Because the data as such are not important. Well, they are important, but not so useful. The usefulness is in what information that they carry. And to extract that information, we use a technique that's called regression. And with regression, we try to sort of summarize what is in the data. And to do that, we need a model. And what is a model? Well, it is a sort of, as I said yesterday, a sort of mathematical representation of how you think that the world is built upon. So it's an abstraction based on a supposed mechanism or a hypothesis that hopefully is science-based. And then you can build such a mathematical model based on the science. But there are also cases where we do not really know what is happening. So you can also think about other types of models, which I call here empirical models, which gives you just a mathematical description. There's not a hypothesis behind it. It just fits the data. And polynomials are one example of that. They are quite frequently used also in science, but you should realize that they are just fits. They are just mathematical fits, and there's no theory behind it. And I think what is important to realize that models are really reflecting our imagination. The imagination should be based on scientific insight, but it is an imagination. It is something that exists in our minds. I don't think models are really there in nature. It is really a human way of dealing with the world around us. And the basic question then for us as scientists is, what is the mechanism that causes the data? But when we collect data, as we also saw yesterday, we always will have variability, errors, and uncertainty, and we need to deal with that. So I took this cartoon from one of well-known food scientist, Ted Labuzza, who, by the way, recently died. But I know him. I invited him also for my kinetic courses a few times. And he once showed me this poem. There was once a chemist in Lata who had terrible data, but he drew a straight line. Now everything's fine except for the terrible scatter. And I think this expresses what most of us will experience when you are going to collect data and you want to do regression. There will be scatter, and how to deal with that. So once again, models describe our belief, our imagination, how a certain process occurs. And for food microbiologists, as many of you are, there are primary models that describe growth or inactivation of microorganisms. There are secondary models that try to explain the effect of temperature, of pH, of water activity, and probably some other factors as well. But you still are using your imagination to describe what you observe in nature. And these models contain parameters like a growth rate or lag times or D and Z values, etc. These parameters are the ones that we are trying to estimate. And that is done by a technique called regression. So what we do is we learn from the data, we try to extract information that tell us something about those parameters. And when we do that, we should not only go for their estimates, we should also go for the uncertainties in those estimates. And this is something that I still frequently see in the food science literature, that people do not report about the uncertainties. And if I ever have to review a paper and I see that, I immediately return it and say, I'm not going to evaluate this unless you give me the uncertainties. I find that very important. Now, let's go back to our very simple example of apples, which we also showed yesterday. So we have collected a sample of apples, we have measured their weight, and we now want to describe that variation. And let's assume for the moment that we can describe it by a normal distribution, which is the red curve here, which is, well, not in contrast to what we actually observe. So perhaps it is a reasonable assumption for this particular sample. So how does then regression work? Well, yesterday you learned about the Bayesian approach, and I have repeated here the Bayes formula. Once again, this is the probability for the data given the hypothesis multiplied by the probability for the hypothesis itself, the prior distribution, and that should be divided by the probability of summing everything together that is possible for this particular case. This probability for the data given the hypothesis is called the likelihood function, and they represent the data, the things that we measure or observe. This prior hypothesis here is called the prior distribution, and both of them, the likelihood and the data, they need to be specified by the researcher. That means us. We have to tell the software what is our likelihood function and what is our prior distribution. The denominator here, the probability for all the data, is something that we can neglect because this sums up everything and it is just a number. I'll come back to that later, but for the moment, we can forget about this factor. So the most important parts that researchers need to specify are the likelihood function and the prior distribution. And if you know those two, or if you assume those two, then you can calculate the posterior distribution, which tells you the probability for a hypothesis or a parameter given the data. This is the way how Bayesian regression works. You have to specify a likelihood function and a prior, and then you can go ahead. The frequent risk regression is much different. This is done by maximum likelihood estimation, usually abbreviated with MLE. And most of you will be familiar with least squares. And least squares is actually a special case of maximum likelihood estimation. And I want you to realize that these two techniques are really very fundamentally different. Even though they lead to the same results in many cases, they are fundamentally different. This one is obtained by sampling. This one is obtained by a mathematical method. Now, how this is done in the Bayesian world is that we need to use numerical techniques to sample from the specified likelihood function and the prior distributions. And that technique is called Monte Carlo Markov chain sampling. I don't have the time to explain all this, and it's also quite complicated to explain. So I refer you here to two nice internet sites in which it is also visually shown how this works. So what you basically do is you specify a sort of sampling space from the likelihood function and the prior, and then the software is going to search in that space for possible values. And some of them will be more likely than others, but this is determined by this sampling technique. And slowly you are building up a distribution. And this is done typically some thousands of times. So it takes time also. It can take minutes and sometimes even hours before you get your results, because this sampling is quite computer intensive. But I'm not going to explain it any further because that takes too much time, and you can look it up on internet. There are many sites that explain how this works, and it is also very well accepted. Then the frequentist regression, the one that you all apply probably. The frequentist regression is completely focused on the likelihood function. So I have written here the normal distribution. So we now assume that our likelihood function is represented by the normal distribution. And what we can do then is we can just vary the parameters. So theta are the parameters, which in this case are mu and sigma. And you are varying parameters and you look at the outcome of your function with the data that you have. The data are represented here by y. And you just vary the theta functions of the variables, and you look at the outcome for the likelihood function, and the one that gives you the highest likelihood is called the maximum likelihood estimate. So that is basically what a maximum likelihood estimation does, looking for the highest likely value for your parameters. And how is this now related to what you all know, or what you probably all learned about the least squares regression? Well, if we accept that there's some disturbing noise at the background, um, if we accept that residuals are normally distributed, then the likelihood of a parameter, if you have only one data point, is given here. This is again the same expression. So y is the data point and mu is the mean, and sigma is the standard deviation. If we have more data points, which we usually will have, we know from probability rules that if the data points are independent, we can multiply the probability function for each data point. So this capital letter pi here is the sign for multiplication. So we can multiply these expressions for each data point. Then what we can do is we can take the logarithm, and as we saw yesterday also, if you take the logarithm of multiplication, you end up with sums. So by taking the logarithm, you get the log likelihood. We now have the sums instead of the multiplication, and these are um, leading to the sums of squares, because that's what you see here. It's the sum of the difference between the data point and the mean divided by the standard deviation, and then that is squared. So that's where the term comes from, least squares. You are minimizing this term here. If you minimize this term, you are actually maximizing the likelihood. So I hope you see that. That's why it's called least squares, because you're minimizing something. So that is the background of least squares regression. It's a special case for maximum likelihood estimation, a special case because you are assuming from the start that this likelihood function is a normal distribution. And this is a big restriction because that's not always the case, as we saw yesterday. And there are quite a lot of assumptions before you can actually apply least squares regression. And I've tried to summarize them here. So one assumption is the residuals, so the difference between the data and the mean, are normally distributed. I'll come back to that in a moment. The second assumption is that there are no systematic errors. That should be always the case, of course, but you have to take care of that. The third assumption is that all the variances around each measuring point are the same. If that's not the case, which can frequently happen, then you can correct for that by using weighted least squares. I'm not going to discuss that, but just for you to know that this is possible. Another assumption is that the residuals are not correlated. So residuals at one measuring point should not be correlated to another one, which in statistical terms means that the covariance should be zero. And we will see tomorrow that there are, in many cases, this is not true, and we have to find another way to deal with that. Another assumption, which is a bit of an open door, is that the model is correct and that the errors that we observe do not depend on the model. We also assume that the x values, so the independent variables, are not containing error. They should be very, very accurate and precise. Only if all these assumptions are fulfilled, then you can assume that least square solution is actually a maximum likelihood estimate. If not, if one of these assumptions is not fulfilled, you may not have the right estimate. And much to my disappointed, most people do not care about these assumptions, but it also means that many results in literature are probably biased because people did not check these assumptions. So you need to check those, and we have several techniques to do that. To check if the residuals are normally distributed, we can use a so-called quantile plot. For the absence of systematic errors, you need to calibrate frequently. You can check for correlated errors by just plotting the residuals against each other, and then you have a so-called lag plot. The errors in X value should be absent. Well, that is again your responsibility to be precise and accurate. Whether or not the measuring points at each X value have the same variance is also something to check. And that can be easily checked by making residual plots. Just plotting the residuals shows you whether they are approximately the same. Then the sample variance, same variance, yeah, you can be done by the residuals. And another thing you have to realize is that if we're going to do regression in the frequentist world, that there is a difference between linear and nonlinear regression. And the difference is that in linear regression really has a unique solution mathematically, whereas nonlinear regression, we really need approximation methods, which leads to more uncertainty. But I do not have the time to explain that, but if you're going to follow my course in kinetics, I'll explain that in much detail because I find that also a very important distinction. Well, then, if you all do that, if you have all these assumptions checked and fulfilled, you can then look at your results, which in the frequentist world are point estimates. But you can also derive the uncertainty in those estimates by looking at the variances and the covariances of the parameters in what is called a covariance matrix. And here is something maybe logical, but also frequently neglected. You cannot estimate parameters independently. If you have more parameters, for instance, if you have a slope and an intercept, they come from the same data, so they must be correlated one way or another. But the question is, how much are they correlated? And that can be found by studying the covariances. And also this is frequently neglected, but it should be done. In the Bayesian world, all of these aspects are covered in the posterior distribution in terms of variances and covariances. You can all derive it from there, but you still have to also check assumptions. Like if you assume that it is a normal distribution for the residuals, you should check that also with QQ plots. Now let's give an example. And I start with Bayesian regression. The notation for the likelihood function is like this. You have to specify a likelihood function, as I mentioned before. And this is statistical notation. You say that we assume that our data y are distributed according to a normal distribution with a mean of mu and a standard deviation of sigma. And then our task is to estimate these parameters from the data. For the moment, I do not have an explanatory value. There's no X value. We just have data, and we want to derive the mean and the standard deviation. And that, if we do that, we have a so-called null model. So once again, likelihood function needs to be specified, but we also need to specify priors. And what could be a sensible prior for the mean? Well, you can, by experience, know that maybe the mean weight of apples will be something around 100 grams. We are not very certain, so we give it a rather big standard deviation. So this means that we are prepared to accept that the mean may be quite different from 100, but we have to specify something. The prior for the standard deviation, I gave that in this case a Cauchy distribution, which is a distribution that has very wide tails. So it also is able to, I give it a value here that gives you also quite a wide distribution, but that's not so critical. But the important part for the prior for standard deviation is that it cannot be zero. So that's why it has a zero here. It means that it is limited at zero. If we do frequentist regression, they usually have another notation. They say I have a model, and that model is augmented by assuming that there are errors. which is usually indicated by the Greek letter epsilon. This epsilon is then assumed to be distributed according to a normal distribution with a mean of 0 and a standard deviation of sigma. So that means on average, errors should be randomly dispersed. And this dispersion is characterized by a standard deviation sigma. So we are not going to estimate epsilon, we are going to estimate sigma. And in the frequentist world, the sigma is estimated by this formula. And SSR stands for the residual sums of squares. So we are trying to minimize the sums of squares, but it will never be 0. And the value that remains after the best fit is called the residual sums of squares. We divide that by the number of data points minus the number of parameters, and we get an estimate of our standard deviation sigma. Okay. Now, just to show you the differences in how you code that, and this is just in R. You can do that in many different ways. You can also do it in Python, for instance, or in any other language. But in the Bayesian way, as I mentioned, you have to specify several things. You have to specify what is your likelihood function. Well, that's indicated here with family is Gaussian. Then I want to do regression for weight. I just have weight. So there's this one here means only give me the mean and the standard deviation. And I have to set the priors. And I mentioned before, I gave a very wide prior for the mean and the intercept. And I give a prior for the standard deviation sigma. Then because it is Bayesian regression, we need also to indicate the sampling. So I propose here that we need 4,000 iterations, so 4,000 times sampling, but we are going to only use the last 2,000. So the first 2,000 samples I throw away, because then the software is still searching for more or less the right area in which to serve. So this is how you specify regression in the Bayesian way. And this last line here is just to store the result. In the frequentist way, you see it is much simpler. We just, and this is the R code, LM stands for linear model. It also says the data are L-star, as we did here, and it says regress weight as such, and that's the same as here. We don't specify any likelihood function here because this is already built in. Yeah, the assumption is that this is a normal distribution. So if you use least squares, by definition, you have a a normal distribution. And that is something to realize. So all these assumptions here are actually built in this code. Also, the prior, you can say, is a completely flat prior. There's no information that we give to the regression procedure. You can say it's an uninformative prior. So it's basically the same, but in the Bayesian world, we need to specify. In the least squares world, we have only the assumptions that are there and need to be fulfilled. So this is the result for Bayesian regression from these apple samples that I just showed about the weight. And what you see here is a comparison of the posterior distribution and the prior distribution. So the dotted line is the prior distribution. So this 100, mean 100 and standard deviation 20, that is this line here. And this is the outcome if we confront that with the data. And then you see that we have a very wide prior distribution, which is now narrowed down to a much narrower posterior distribution, which means that we have decreased our uncertainty or increased our certainty, if you like. So now we can see that the mean that we find in this case is 88 grams. And we see also the uncertainty in that estimate. So we visually see now how uncertain we are about our estimate. And the same goes for the standard deviation. The standard deviation can only be positive, so it is slowly decreasing. but confronting it with the data shows that our initial guess for the prior is not completely right, but that is not a problem because the data are so numerous that we can safely say that the standard deviation is around 16 grams. And you see here the distribution. You also see that it is a little bit skewed. And this is due to the fact that standard deviations cannot be negative. So they're always extending to the right. But the interesting part here is that the standard deviations, which characterizes the variation, it's itself also uncertain. And how much the uncertainty is, we can read again from this posterior distribution. These bars here are the median with the 95% confidence interval. You can also express that in numbers, but personally I find the distributions much more convincing. If we look to the frequentist result, then we see the same formula, weight versus the one with the same data, but now we see an output for the residuals. We see an estimate of the intercept. That's the same as we saw before, 88 grams. And we also see the standard error, which is 16, which is also the same as we found with the Bayesian result. But you do not see an uncertainty estimate here on the residual standard error. It's just a point estimate. We do see an error for the estimate of the mean of the apples weights. We also see here our p-value, and this p-value is very, very low. And this p-value refers again to the zero hypothesis, the H0, the null hypothesis. And what this number says here, the probability that the zero hypothesis is true, and remember the zero hypothesis always there is no effect. In this case, it would mean there's no reason to assume that the mean is 88 grams. Well, that probability is very, very low, and therefore we conclude, okay, it is definitely 88.1 because the probability for something else is very, very low. This is how you should interpret p-values. Now, this was, of course, a very simple example, and I want to move now from a null model to a more realistic model for what is called an explanatory model. We want to explain why, for instance, weight or size of apples depends on weight. So in the null model, we have, I told you, we have assumed that it is a normal distribution of all the data points around a mean. If we are going to extend our model by introducing explanatory values, so predictors, we are going to need a model. We need to put forward how we think that a response Y is linked to a predictor X. So for instance, bacterial growth could be Y and the predictor could be time. So you can forward a model. And then you need to confront that model with data. Now, let's say that, again, this is all very schematic. So, let's say we have done some experiments. We have varied an X value. It does not really matter what X is at this moment or what Y is, but we have measured something that looks like this. And what we see is that the Y value tends to increase with the X value. And we want to know now what is the relation between those two variables. So, for instance, how is apple size related to apple weight? The assumption of normality of data that we already saw at the null model, we can also make that assumption for each measurement, but you need to check it afterwards if it is correct. But then you can still estimate sigma, even if you have only one measurement at each X value, you can still measure that because it's usually assumed that sigma is constant. So we still have a few measurements from which we can derive sigma. So if we now for the moment assume that this increase of y with x is linear, we can draw a line which we can represent mathematically by the equation for a straight line, a plus bx, in which we call a the intercept and b the slope. Then we see that this line is surrounded by data points, but not exactly corresponding to the data points. So we do see variation here again. And what we learned yesterday, we can describe that variation by a distribution. So that's what I try to show here. At each x value, we assume that data points around the line are represented by a normal distribution. And even if we have only one data point, we can still assume that. Now, there are some concepts that I like you to think about. So the line that we see here is the expected value or the mean for each value y at a value x. That's the model. And we call that the expected value or the mean. We assume this normal distribution, which we then can characterize by a standard deviation sigma. And what we also have is the deviation between the measured point and the mean, which we call the residual. So the mean is called the expected value. And I like you to note now that first we had one mean, but now we have a mean at each x value. We call that mu y. And also now our model is mu y is represented by this equation for a straight line. And each y corresponds to the specific x value. The standard deviation sigma I we have assumed to be constant. So we assume that every deviation at each xy point is the same. That is usually assumed, but frequently not specified, but you can check it. And if it is not the case, you can actually relax that assumption. But I'm not going to explain that now because of time, but it is possible to correct for that. But in most cases, people just assume that this standard deviation is the same, and that's not always correct. And then we have this residual, the difference between the mean and the data point. And I also would like you to realize that this residual is not the same as a standard deviation, because the residual is actually the difference between the measurement and the mean, but the standard deviation is a characterization, so an estimate of all those residuals. Once again, if we do Bayesian regression, you need to specify a likelihood function. Well, we do the same as we did before, but now we have replaced our overall mu by this mu y, so the mu at each x point. And we can then replace the mu y by this model, because that's our assumption that the mean of each x value is represented by this model. It could be any other equation, but for now we assume it's a linear model. And then we need, again, as usual, to specify our priors. We have to say, what do we expect for this intercept A? We can start by giving it a normal distribution with a mean and a standard deviation. And the same for the slope. We can give it a standard deviation and a mean and assume it is also normally distributed. We also have to specify the standard deviation. Well, in this case, I have given it an exponential distribution, which also gives only positive results. And here is the code again to show that it is basically simple, but you need to specify. Again, we assume a normal distribution. Now we are going to regress the diameter versus the weight. And the one means we want an intercept. We have to specify our priors for the intercept, for the slope, and for the standard deviation. We also have to specify how many iterations we want to do in the Markov chain Monte Carlo sampling. And here below is the frequentist specification, which once again shows the same formula, diameter versus 1 plus weight, the same as here. And the data is L star, which is also the same as here. But for the rest, there's no specification because it is assumed it's a normal distribution and the sigma are the same. So this is the result of regression, of doing the linear regression of size of apples versus their weight. And we can find the estimates for the parameters, which I listed here. These are the numerical estimates from the Bayesian results. And here you see the graphical representation of the distributions. So once again, you see that the prior is very weak. It only gives a hint, but when you confront it with the data, it becomes much more clear. So we have updated our knowledge, you can say. The same goes for the slope. This is the slope, this is the intercept, and the same goes for the standard deviation. And these numbers here are just derived from these distributions. And we can derive standard errors and we can derive confidence intervals if you like. If you do the frequentist results, you get the same numbers. So you get the intercept, also 40, and the slope is 0.224, and you get a standard deviation of 0.82. You don't get an error on the standard deviation. You do get an error on the intercept and slope. And again, here you have the p-values, which also indicate here that they are significantly different from zero, which is the null hypothesis. There's no effect. But our p-value tells you that it's very, very unlikely that there is no effect. So that is the difference between the Bayesian and the frequentist results. With the Bayesian result, you get much more insight in the behavior of your parameters. With the frequentist results, you just get numbers. Then I also talked to you about parameter correlation. And this is, again, a Bayesian result where this is all derived from the posterior. So that's what I said before. You can do all kinds of calculations with the outcome of the posterior. And here you see the posterior distributions again for the parameters, but also the correlations between the two. And you can see that the two, the slope and the intercept, are quite strongly correlated, 0.98. Whereas the slope and the standard deviation are not correlated, and also the intercept and the slope and the standard deviation are not at all correlated. Is this bad? Not necessarily, but it's a warning that you should be careful if you're going to do calculations, because a strong correlation means that they are strongly dependent on each other. And there are ways to avoid that, but there's no time to explain how that works. These figures that I showed for the Bayesian regression cannot be obtained from frequentist regression, but we can obtain so-called covariance matrix for all the parameters. So if you have parameters A, B, and C, you get covariances and variances. So this is a so-called covariance matrix where the diagonal indicates this variances for the parameters, and the off-diagonal elements here are telling you about the covariances between A and B or A and C. And these will be just numbers. And these numbers do not tell you much, so therefore one usually recalculate this in so-called correlation matrix, where, of course, at the diagonal now you only see once, because B is correlated with itself perfectly, of course. But on the off-diagonal elements, you now see a correlation coefficients between the parameters. And as a rule of thumb, if you see numbers that are below 0.8, you should not worry. If they are above 0.9, well, then you have to be careful and start to think about, is this a problem, yes or no? So parameter estimates regression leads to parameter estimates and residual variance. That should always be reported. But also, and this is frequently neglected again, variances and covariance, because they tell you about the behavior of your parameters. And parameters are always correlated. And this correlation is particularly important if you're going to do calculations with your parameters. So if you are going to do risk assessment, for instance, and your parameters are strongly correlated, that may have a big effect. So unfortunately, not all the software packages give this information, and especially Excel does not do that, which is really a shame, because I find it really essential. Now there are ways with macros in Excel to get them, but it should be their standard. Then residuals should be checked, which is just the difference between the mean and the values, and they should be randomly dispersed. And you should also check a quantile plot, and they look like this. So the theoretical behavior is this line, and your data should giggle around it. And in this case, this is quite nicely. So you can test your assumptions that of normality, and that seems to be okay. Okay, I think this is a good point to have a small break of 5 minutes. So shall we come back again at 10 o'clock, and then I can finish with some final notes about these basic assumptions of regression. Is that okay?
Yes, it's perfect. So
Okay.
we'll see each other again at 10.
Okay, see you then.
You. Also, for the participants, if you have any questions, type it in the Q&A document in the chat.
Well, on my clock it's 10 o'clock, so shall I start again?
Yes, please continue.
Okay. Okay, so far I have only shown a linear model, and I want you to realize what that actually means. It means that it is additive. You have proposed a model in which effects are additive. So I've given here two equations, um, and both are additive and therefore linear model, even though this equation here is describing a nonlinear line. So nonlinear, if you have a nonlinear model, it means that they are not additive in the predictor. And here is one example, an exponential model or a Michaelis-Menten type model. Those are not additive and therefore non-linear. And also in microbiology, most models are non-linear. Linear models therefore do not mean a linear relation between x and y. It means just that they are additive. So also this kind of curves, which is a polynomial, you could analyze with linear regression, even though the relation itself is not linear. We also can derive confidence and predictions intervals when we have parameter estimates and in their uncertainties. So the difference between the two is the following. If you want to calculate confidence intervals, this tells you something about the expected value of the mean. In frequentist world, if you would repeat the experiment many times, what is then the how many times will you find the mean back in your interval? And this 95% confidence interval is determined by the uncertainty in the parameters. So I showed you the standard deviations or standard errors of parameters. Those determine the width of such a confidence interval. Then we have prediction intervals. There is not about the mean, but it is about what are the expected values of new measurements if you would repeat the experiment many times. And this is also determined by the uncertainty in the parameters, but again, also by the residual variance or residual standard deviation. So that can make quite a difference depending on your outcome. And by the way, 95% is just arbitrary. It could also be 90% or 89%, doesn't matter. It's just what you define. So this confidence interval is really about retrodiction. So you are using the data and then you are going to retrodict what you would expect for the mean. And the prediction interval is really for new observations that are not yet there. And here is an example of that. Again, for this apple example, the dark blue ribbon here is the confidence interval for the mean. And you can see that that is much narrower than the confidence or the prediction interval for new values. And I think both intervals make sense to display. Now, looking at again at the difference and similarities between Bayesian and frequentist regression, as I have showed you, the software needs specified prior distributions and likelihood functions. And as a bonus, you get the complete posterior distributions showing all the uncertainties and possible further calculations can be done. And in the Bayesian world, it does not really matter whether you have a linear or a nonlinear model. It is because the sampling takes place from the model that you specify, and the software doesn't care whether or not it is linear or nonlinear. In the frequentist world, you don't have priors. You always, if you use least squares, you always assume it is a normal distribution of residuals, and you only get point estimates. The uncertainty that you can calculate with confidence intervals are not probability distributions. And very, very importantly for frequentist regression is that there is a different procedure for linear and nonlinear models because the way it is calculated, it's much different from the Bayesian world. So linear models can be derived exactly by mathematical procedures, but nonlinear models need approximations. But again, there's no time to explain that. I just want you to be aware of that. I also think, and I hope I have convinced you that with Bayesian results, you get intuitive results. There's no need to talk about p-values. You just have to decide for yourself. The disadvantage is that you need some programming skills to get it going, but I hope I showed you also that it's basically very simple. Depending on the complexity of your model, it can take considerable computer time. And just by changing the likelihood function, you can go away from normal distributions. You can also use Poisson or log normal or whatever you think is suitable. That is just by changing the likelihood function without any problem. The frequentist world, if you do frequentist regression, you don't need programming skills. You can just click and run and you will get an output almost immediately. But you will not have probabilities for models and parameters, and it is frequently misinterpreted. I don't know if you know about this phenomenon of p-hacking, that people are designing their experiment in such a way that there will always be a significant p-value. That is a big danger with the frequentist approach. Well, as I said, Bayesian regression is not limited to the assumption of normality. You can do it with other likelihood functions. But you should note that this is in principle also possible with maximum likelihood estimation. But if you do least squares, if you choose for least squares, you are automatically related to a normal distribution. But if you want to do Poisson, for instance, you have to use maximum likelihood estimation. Now I want to finish this part by telling you a little bit more about other likelihood. And in this case, I chose for Poisson regression because that is useful for microbiology. As I also explained yesterday, in microbiology, you have counts, plate counts, and that means that you have integers that are always positive. And especially if you have low plate counts, that could be quite important. Well, the Poisson distribution has only one parameter, which estimates both the mean and the variance. And if we change from a normal distribution to a Poisson distribution, the goal remains the same. We want to estimate this lambda, this parameter, from the count data. And the big advantage of Poisson is that you can also include zero observations. But the downside is you have to make sure that lambda, the parameter lambda, cannot be negative. So how does that relate to what we saw for normal linear regression? So in normal linear regression, we assume a normal distribution and we replace this mu I by a model. Here it is a linear model, but it could also be a nonlinear model. And then we can estimate the parameters, alpha, beta, sigma, whatever you have. In Poisson distribution, we cannot have negative models, so negative parameters, because the counts are always positive. So to prevent that the model is going to predict negative values, we have a method to do that by taking the exponent of the model. And then this parameter of the Poisson distribution is replaced by the exponent of this linear model. And you can also take this the other way around. You can take the logarithm of this lambda. So then what you do is you say that your data are dispersed according to a Poisson model, but then the log of that parameter is linear related to this model. And if you do it in this way, which is frequently done, people do not use it like this, but like this, but it's basically the same. And remember, the log is the natural logarithm here. This transformation is called a link function. So it links the parameter of a distribution to a linear model. And this is called a generalized linear model, and Poisson regression is one example of such a generalized linear model. Another example that you may have heard of is logistic regression, where you use a Bernoulli distribution, where the link function is the logistic equation. And don't confuse that with the logistic equation that is used in microbial logistic models, because that's not about regression, that's really the model itself. And GLMs, generalized linear models, are especially developed to handle non-normal data. So it is represented here from a paper that I really could recommend to read. It's not about our field of research, it's about linguistics, but it explains very, very well how Poisson regression actually works. So the assumptions are all the same, but the mathematical implications are different. So once again, if you have a normal distribution, you just replace the mean by this model. If you have logistic regression, you replace this probability by this model. If you have Poisson distribution, you replace its parameter by the exponent of the model. Now, there is a potential problem with data if you want to use the Poisson model, that the mean is actually not equal to the variance, which is an assumption in the Poisson model. But the data may tell you different. And if that happens, it's called over-dispersion. So if the variance is actually bigger than the mean. Now, of course, there's a solution for that. You just have to modify the Poisson distribution a little bit, and then it's called the Poisson gamma, or also called a negative binomial distribution, which includes an additional parameter to account for this over-dispersion. And I think this is probably the better way to treat microbial data. And I just want to finish by showing you that this approach is now coming up more regularly in the literature. You can now see more papers appearing that use this approach with the Poisson and the negative binomial regression, where they use this log-link function that I showed you. And you can also wonder why not take the logarithm of the data as we usually do in microbiology. Well, the difference here is that if you do this, take the logarithm of the data, you are transforming the data. If you use this log-link function, you are not transforming the data, you're only transforming the parameter. And that is in principle better because you use the counts directly. And also you can use zero counts, which is not possible if you take the logarithm of zero counts because that does not exist. So in that sense, it's also from a theoretical point of view better to use this log-link function than taking the logarithm of the data. And I found a nice paper that also gives this message. It's about ecology, not about microbiology, but it gives also clear arguments why you should not log transform your count data. Now, as I said, I see more and more papers using this approach. So this one is from Hiura et al. I think from 2019, where they use this generalized linear model. And this is one of their figures in that paper. Here they have taken the logarithm of the data and assume a normal distribution for the data, for the log of the data, I should say. And here they have taken the negative binomial negative binomial distribution. And you can see that this distribution of uncertainty is very different from the normal distribution. Another one is from Alberto Garra, who was a postdoc at our microbiology lab in Wageningen. Marcel Zwietering was the professor of food microbiology, and myself, where we also used this negative binomial to estimate low plate counts, and we have called that the most probable curve method. There's no time to explain all this, you can look it up yourself. But what we did is we also compared the different types of approaches. So this is what most people do. You take the log of the data and you assume a normal distribution. You get this kind of plots for inactivation. If you would take a Poisson distribution, it's completely wrong. And this is because the mean and the variance are not the same. So you get completely different fits that make no sense at all. This is the variability if you take the negative binomial, which also gives very, very wide prediction intervals. And this is what we came up with, the most probable curve method, where we actually used the negative binomial, but in a different way, to come up with a prediction of the uncertainty in our regressions. And as you can see, it nicely tells us that the uncertainty increases with lower plate counts. Then there is a very recent paper from 2025, which also tells about these things. I think this was a very nice overview where they show all kinds of different situations with growth and inactivation, also trying to distinguish between technical and biological variability. So I would highly recommend to read this paper to see the applications of Poisson and negative binomial regression in food microbiology. Then finally, I have supplied you with 3 tutorials which explain in much more detail linear and non-linear regression. And I used the same data for each tutorial, but I used different software. So the 3 are completely the same in terms of the data, but they are different in the way the data are analyzed. And you can look at the differences yourself. This one is in Excel, where most people are probably aware of how that works, but it's done in the frequentist way. I also did it in R with the frequentist way, and I did it in R with the Bayesian method. So you can compare the differences. And maybe you can use those tutorials as a blueprint for your own problems. It also tells you a little bit about how to install the software that you need, especially for Bayesian regression. You need some special things. I did not. This was not. How should I say that? It's not. I did not have examples yet with this Poisson or negative binomial regression. So it's based on normal distribution assumptions. But as I mentioned, especially in the Bayesian method, it's very easy to switch to another likelihood just by changing the family. And you can go ahead again. So I hope that is useful for you. Feel free to distribute it if you like. They're open. You can also ask me for comments, or if you have questions, feel free to approach me. I hope it is useful. And if you have feedback, I like to hear that as well. So that was what I wanted to share with you today. And perhaps there are some questions left.
Yes, Professor, thank you so much for joining us once more and sharing all this valuable knowledge with us. We have a few questions, and I will start. Also, before I start with the questions, I have a quick message for our attendees. Please take a moment to complete the form that Natalia will share in the chat. This is necessary for us to issue your certificate. So let's move to the questions. Okay. The first one that I saw here is, what types of prior distributions are commonly used in Bayesian linear regression?
Yeah, nice question. Priors are usually considered as a difficult topic because people do not really know what they should propose. My advice would be usually a normal distribution for the priors is doing well, except for some parameters that you know already that they cannot take on certain values. So, for instance, if you have rate constants or if you have d values, you know that they cannot be negative. So, in that sense, you can choose a log normal prior or a exponential prior to make sure that the software will never go into the negative area. With a normal distribution, that is possible in principle. However, if you assume a normal prior, if you, yeah, go ahead.
No, I don't know.
Oh, okay, it stopped. But if you feel free to ask questions also live, that's possible. But let me finish my answer. So I would always start with a normal distribution, and what always helps is to plot the prior, just to see how it looks like. And what you also should realize is that the prior should reflect your knowledge. So maybe you know already a lot about a certain parameter, and then you can give it a narrow prior. But if you are very uncertain, you can also give it an uncertain, a big standard deviation to express that uncertainty. So a prior is really your choice. But what I really like about this approach is that you need to specify it. And people may disagree with your choice. So you have always to, if you make a publication, you have to specify why and which priors you used. And people may disagree and do their own result with another prior if they like. And that's fine because that is open science.
Yeah,
Does
that's true.
that answer the question?
I think yes. I don't know if anyone from the audience wants to clarify something on this. I think no. Another question is, how can Bayesian regression be applied to predictive microbiology models for microbial growth or inactivation? And can we still use the same models?
Yeah. Yes, the models do not change in the frequentist or the Bayesian world. They are exactly the same. But the interpretation becomes different for the parameters. And especially if you ask about predictive modeling, then I think the Bayesian outcome is much more sensible because you are now using the whole distribution. And so you can, as I mentioned, this posterior distribution contains a lot of numbers that you, so thousands and thousands of numbers that together form a big source of information. And you can use all those numbers at the same time to calculate predictions in terms of how uncertain will be your calculations. So these prediction intervals that I showed to you are much more clear with posterior distributions. I did not show how you actually calculate the prediction interval in the frequentist world, but it is very difficult to explain why that formula is there. Actually, I do not understand it, but it works. So in that sense, you could use it. But with the posterior distribution, it becomes so logical to say, well, I have this uncertainty in my parameters. And now if I want to calculate that, for instance, for risk management, I can use all that uncertainty in my risk management calculations. So it is much more intuitive. But the models remain the same. So the models do not change because of frequentist or Bayesian. You can use them in both approaches.
Okay. I see here that, okay, if we want to compare 3 or more different parameters simultaneously, what statistical model you would recommend for analyzing and interpreting such data effectively?
That is really depending on the type of model that you use, but and the data that you are going to have, because there should be enough information, of course, in the data to extract information about the parameters. So if you have very few data and many parameters, then the parameters will be very uncertain and probably also strongly correlated. So that means that you do not get much information. But again, compared to the frequentist world, you need less data in the Bayesian approach than in the frequentist world, because in the Bayesian approach, you can use the knowledge that you already have in the prior. But in general, you can estimate many more parameters at the same time. Last year, I was co-supervisor of a PhD student who worked in toxicology. And I think she had something like 20 parameters in her model. And that is still possible in the Bayesian world. So that is not a limitation, but it depends really on the data that you have. Is there enough information in it?
Okay, and we have now a question in the chat. I see Sulev. I don't know if he wants or she wants to do the question, but I see here that it says in microbiology, we calculate CFU by considering between 30 and 300 colonies in Petri dishes, but sometimes we have over than 300 colonies. Can we use Poisson regression to calculate prediction?
Yes, that's a good question. Yes, it can, but I refer to this, one of the papers that I showed in my presentation. I think the last one from 2025 does consider this possibility, and otherwise it will be in one of the other references, because I have seen this tackled this problem by well, it will not be Poisson regression, but negative binomial regression or Poisson gamma regression. But I think in principle it is possible. So this problem has been considered in the literature.
Another question is there a null hypothesis for the Bayesian approach? And can a Bayesian regression assume a Poisson distribution?
The last question, yes, you can just specify Poisson distribution as the likelihood function. That is not a problem at all. And then the first one was, can you specify the null hypothesis? Yeah. Well, the Bayesian world is not so much about a null or an alternative hypothesis. You can have many hypotheses at the same time. And that's another advantage. So you can say, I have one hypothesis and the second and the third, and then you can just compare them in terms of their probabilities. So, for instance, you will find that hypothesis 2 has 90% probability, and hypothesis 3 only 50%, and hypothesis 1 maybe 65. And then you can choose based on that hypothesis, on those results, that hypothesis 2 is the most likely one. But it does not mean that you should reject the other ones. That is the basic problem with these p-values. I think a lot of hypotheses are rejected because of a very stupid number. It's much better. A hypothesis that had a p-value of 0.1 could still be a very interesting hypothesis. But just to reject it because it has a high p-value, I find very stupid. And in the Bayesian world, you cannot do that. You just have to state, I find hypothesis 2 more probable, and therefore I think that is the most likely one. But you cannot say hypothesis 1 is not valid. It is valid, but is less likely. So the habit of comparing H0 and H1 is not done in the Bayesian world.
Okay, and I think we have time for one last question. And that is, what if we are not sure about the values of the priors? How do the priors affect the final outcome?
Yeah, that is a very good question, because that is usually the criticism that that people have. To say, well, you can choose a prior and therefore influence your results. And that is indeed a danger. And that is why I also find that people should publish the prior they have used so that people can check this. But there is a very simple way to check that, and I also showed that in my presentation. You can just plot the two distributions, and the ones that I showed to you, you could see that the prior was very wide and the posterior was very narrow, which means that the data are determining the posterior. But if your prior would be very, very influential, you would see that the posterior is turning towards the prior. And if that happens, you have to be careful. You have to be to make sure that your assumption of a strong prior is really necessary. So it is you always should compare the priors and the posteriors to see whether or not they are dominating. And this way of looking at it is called prior sensitivity analysis. So that's why I'm in favor of always plotting distributions to see what is happening.
I see now one more question in the chat. Maybe we can ask it. It's, would it be possible to apply what is this? Ones of this model to predict ones of these models to predict biofilm formation?
Well, that really depends on the mechanism behind biofilm formation. So if you have a model that describes that, then you can do it. But as I tried to explain before, it is your imagination, it's your responsibility as a researcher to think about the data that you are collecting. What is the mechanism behind that? And if you have a mechanism that describes biofilm formation and you are convinced that that is the way it works, yes, then you can use it. You can use any model that you propose as long as it is based on science. So definitely, yes.
Okay, that's clear. Thank you very much again for all this knowledge, for the discussion, for your time. We are really happy to have you here with us. And we have also one more day. If someone wants to come back tomorrow to something to ask, we are open.
Yeah.
Thank you again. I don't know, Federico, Natalia, is there anything we have to mention?
Thank you so much, Professor. And see you all tomorrow at 9 a.m. for our last session.
Okay.
Yes, thank you.
Bye-bye.
Okay,
Bye.
thank you. Thank you.